Welcome back to the Deep Dive. Today, we are tackling, well, what I think is probably the most complex and certainly the most rapid biological transformation in human life.
It's quite the journey.
Absolutely. Our mission today is nothing short of total mastery of the first week of human development. We're going way beyond the textbook summaries. We're diving deep into the specific chronology, the precise hormonal signals, the exact cellular pathways, and all those molecular mechanisms that basically bridge the gap from a single ovulated cell to a blastocyst ready to implant.
That's absolutely the goal. The source material we're working from today is, well, it's clinical level embryology. It really focuses on the how and the why.
Right.
So if you're looking to truly grasp the systems, ensuring these earliest moments are successful, you've got to pay close attention. The hormonal controls, the cellular sorting, the critical timing, it's all essential.
We're aiming for that deep professional understanding.
Exactly. Mastery.
I love that. Mastery of the complex chronology. Okay, so let's unpack this incredible journey, starting right at the beginning of the whole cycle. And the starting point isn't actually the ovary, is it? It's the brain.
That's right. It's all governed by this perfectly orchestrated hormonal cascade.
Yes.
Yes, the entire regulatory axis kicks off in the hypothalamus. Think of it as the master conductor.
Okay.
It secretes gonadotropin-releasing hormone, GnRH, but it does so in pulses. That's important.
Pulse release. Got it.
Then GnRH travels down to the anterior lobe of the pituitary gland, a structure sometimes called the adenohypophysis.
Right.
And that stimulation makes the pituitary release the two big players, the gonadotropins, follicle-stimulating hormone, FSH, and luteinizing hormone, LH.
FSH and LH.
Those two are the real drivers. They dictate the rhythm, the function, everything about the regular monthly cycles.
It's just incredible that the whole system starts with a signal from the brain. Now let's focus on FSH early on. The source material highlights something a bit subtle but really critical about when FSH is actually needed for follicle growth. It's not about starting things off, but more about selection.
That is a key insight, absolutely. At the beginning of each cycle, you have this pool, maybe 15 to 20 primary stage follicles or preantral follicles.
Okay, 15 to 20.
These have actually already started developing from the primordial stage, and that initial development doesn't depend on FSH.
Oh, okay. So FSH comes in later.
Exactly. FSH's job at this point is really one of rescue. Without that FSH signal, or if they don't respond properly, those follicles just undergo atresia. They die off.
So FSH saves them.
It rescues a select group, yes.
Right.
A cohort of already developing follicles are rescued from degeneration, and FSH promotes their further growth.
So the journey from primordial to primary is kind of slow, constant, happening in the background. But then the jump from primary onwards is this high-stakes competition driven by FSH.
That's a great way to put it. High-stakes competition.
And even with the rescue team, the competition is fierce. Out of those 15 or 20 contenders, usually only one makes it.
That's the normal physiological result. Yes, just one follicle reaches full maturity. It becomes the dominant one, the one that will actually release an oocyte.
And the rest.
The others, well, no matter how far they got, they degenerate. That process is called atresia.
Atresia.
And when that happens, the oocyte itself and the follicular cells around it, they're replaced by connective tissue. It leaves behind this little fibrotic scar called a corpus atreticum.
A corpus atreticum.
Right. So it means the vast majority of potential eggs a woman has are actually eliminated through this ongoing, really rigorous selection process.
Wow. Okay, let's follow the winner then, the maturing follicle. As it grows, it becomes this highly efficient hormone factory, two different cell types working together to pump out estrogen.
It's a classic example of endocrine teamwork. Neither cell type can do the whole job alone. First, you have FSH stimulating the maturation and, importantly, the proliferation of the inner follicular cells, the granulosa cells.
Granulosa cell.
And this proliferation isn't just random, it's mediated by a specific signal molecule. Growth differentiation factor 9 or GDF9.
GDF9.
Yes, GDF9 is part of the transforming growth factor beta, or TGFB family. It's absolutely crucial for keeping those granulosa cells healthy and dividing.
Okay, so GDF9 gets the machinery going.
Yeah.
But where do the actual building blocks for the hormones come from?
They come from the layer right next door, the theca interna cells.
Theca interna.
These cells produce the necessary precursors, specifically androstenedione and testosterone. These are androgens.
Right, male-type hormones.
Exactly. These androgens then diffuse across the dividing membrane into those highly active granulosa cells. And the granulosa cells have the key enzymes, the aromatases, to take that androstenedione and testosterone and convert them into the really potent estrogens. estrone, and the main one, 17-beta-estradiol.
Such an elegant two-cell system.
Yeah.
And the estrogens produced, they're not just acting locally, right? They send signals system-wide, priming the body for a potential pregnancy. Our sources list 3 critical effects.
Yes, 3 effects that really have to happen together almost synchronously. First, the main target organ, the uterine endometrium.
Theory of lining.
The estrogen drives that lining into what's called the follicular or proliferative phase. It's actively rebuilding the tissue that was shed during the last period.
Okay, rebuilding the lining. What's second?
Second, and this is absolutely crucial for fertilization success, estrogens act on the cervix.
Yeah.
They cause a really dramatic thinning of the cervical mucus.
Making it easier for sperm to get through.
Precisely. It essentially opens the gate, makes it physically possible for sperm to travel from the vagina up into the uterus.
Okay. Lining rebuilds, mucus thins.
Yeah.
And the third effect, it sets up the next big event, right?
Exactly. The third effect is a positive feedback loop. Those rising estrogen levels stimulate the anterior pituitary gland to release a sudden, very high level of LH. The luteinizing hormone surge.
The LH surge.
And that surge is the immediate trigger for ovulation and the final maturation steps of the oocyte.
Okay, we've reached the peak moment, the LH surge. Just before midcycle, FSH and LH have worked together, expanding that vesicular follicle to maybe 25 millimeters, making it the mature vesicular or Graafian follicle.
That's right, the Graafian follicle.
But it's this sudden, almost explosive increase in luteinizing hormone that dictates the timing of release and those crucial final steps for the oocyte.
The LH surge is an incredibly potent signal. Its abrupt arrival causes this rapid increase in the levels of something called maturation-promoting factor inside the oocytes.
Maturation-promoting factor, MPF.
Right, and this factor is like the chemical switch that forces the primary oocyte, which has been stuck in meiosis IT since before birth,
Arrested development.
to finally complete that first meiotic division.
Okay, so meiosis IT completes. That gives us the first polar body and the secondary oocyte. But the source material says it doesn't just finish meiosis right away. It stops again just before release. Why start meiosis the second only to arrest it again?
It seems to be a mechanism for, well, efficiency and preservation. The LH surge starts meiosis the second, but it halts the process specifically in metaphase.
Metaphase arrest.
And this arrest is critical. The secondary oocyte is now essentially ready, genetically poised to receive the male pernucleus upon fertilization. But by delaying the final completion, the oocyte conserves its limited resources and protects its genetic material.
Okay.
This arrest happens roughly 3 hours before ovulation itself, so the released oocyte is genetically prepped and metabolically optimized for immediate fertilization.
So the LH surge is driving the chemical maturation, but at the same time, it's driving the physical rupture of the follicle. This sounds less like a gentle release and more like a controlled demolition.
That's actually a pretty good analogy. First, you see physical signs. The ovarian surface bulges out locally, and a vascular spot, kind of pale, appears at the very peak that's called the stigma.
The stigma.
Then, the high LH concentration triggers the release of an enzyme called collagenase.
Collagenase breaks down collagen.
Exactly. It starts literally digesting the collagen fibers that hold the follicle wall together. And at the same time, the surge causes a big increase in prostaglandin levels.
Prostaglandins, like inflammatory mediators.
Yes. They act locally to trigger powerful muscle contractions in the ovarian wall right near the stigma.
So it's the combination, the enzyme weakening the wall and the muscle squeezing that pushes the oocyte out.
Precisely. The oocyte gets extruded, still surrounded by those granulosa cells from the cumulus oophrus.
Cumulus oophrus.
Once it's free, floating briefly in the abdominal cavity, the cumulus cells that remain stuck to the oocyte's protective shell, the zona pellucida, rearrange themselves. They form this radiating crown called the corona radiata.
Corona radiata. Okay, the oocyte is free, wearing its crown, but now it needs to be caught. If the timing's off or the collection fails, it just gets lost in the body cavity.
That can happen, yes. But normally it's an active and quite sophisticated collection process. The fimbriae, these finger-like projections at the end of the uterine tube.
The fallopian tube.
Right, the oviduct. They start sweeping over the surface of the ovary, and at the same time, the rhythmic contractions of the tube itself increase.
So the fimbriae sweep it in.
They guide it in, yes. The sweeping motion, combined with the beating action of cilia lining the tube's epithelium, draws the oocyte into the tube's opening.
Okay, it's captured. Now, once it's inside the tube, the environment changes, And those cumulus cells, the corona radiata, start to detach.
Yes, once it's safely inside, those cumulus cells gradually lose contact with the oocyte and the zona pellucida. Propulsion now relies on internal forces within the tube.
Like what?
Two things, mainly.
Yeah.
The peristaltic muscular contractions of the tube wall, sort of squeezing it along, and the constant beating of the cilia in the lining, pushing it forward.
And the speed isn't constant.
No, crucially, the rate of transport is regulated by the overall endocrine status, the hormone levels. Our sources say it takes a pretty precise window, approximately 3 to 4 days, for the fertilized oocyte to reach the uterine lumen, the main cavity of the uterus.
3 to 4 days. And that timing isn't random, as you said. It's vital to ensure the embryo arrives exactly when the uterus is ready, prepared by progesterone. We'll definitely get to that. But before we leave ovulation, what about clinical signs? Can women feel this happening?
Some women do experience Mittelschmerz.
Mittelschmerz.
Yeah.
German for middle pain.
Exactly. It's often described as a minor, sharp pain on one side around midcycle. It's thought to be caused by the follicle rupturing, maybe a tiny bit of bleeding into the peritoneum.
And temperature changes.
Yes, more broadly applicable is the rise in basal body temperature. The surge of progesterone that happens right after ovulation causes a slight but measurable increase. Charting this is a reliable way for women to track their cycles and time conception.
Okay. And what happens when this natural system fails? If gonadotropin levels are too low to trigger proper follicle development, we can step in pharmacologically.
Yes. If women are anovulatory not ovulating because of insufficient gonadotropins, we can administer drugs to stimulate their release or activity.
Like Clomid, which we mentioned earlier.
Right. Drugs like clomiphene citrate are effective, but they often cause the stimulation of multiple follicles rather than the usual single dominant one.
Ah, so that's why fertility treatments increase the chance of multiples.
Exactly. This artificially induced multiple ovulation significantly increases the risk of multiple pregnancies, twins, triplets, sometimes more by up to 10 times compared to natural conception rates. It really highlights how tightly regulated the natural selection of just one follicle normally is. #AV. The corpus luteum and uterine preparation for implantation.
Okay, the oocyte is released. Back at the ovary, the ruptured follicle isn't just discarded. It undergoes this rapid, really essential transformation into the corpus luteum. This is the structure that tells the uterus, hey, potential pregnancy incoming. What drives this change?
So the granulosa cells and the theca interna cells that were left behind, they get rapidly vascularized. Blood vessels quickly grow into the collapsed structure.
Okay.
And under the continuing strong influence of LH, these cells undergo a process called luteinization.
Luteinization, becoming lutein cells.
Precisely. They start accumulating this characteristic yellowish lipid pigment, hence luteum meaning yellow. They become lutein cells, and this whole mass forms the corpus luteum. Its main job now is to massively ramp up the secretion of estrogens, but even more critically, progesterone.
Progesterone, the hormone of preparation. It switches the uterine lining from that estrogen-driven rebuilding phase, the proliferative phase, to the receptive secretory phase. Let's quickly recap the uterus structure just to orient ourselves.
Sure. The uterus wall has 3 main layers. The outermost is the perimetrium, that's just the peritoneal covering.
Okay.
Then the huge middle layer, the myometrium, which is all smooth muscle, that's what contracts during labor.
Right.
And then the dynamic inner lining, the endometrium, that's the mucosa. And it's the endometrium that cycles every month from puberty or menarche until menopause, completely driven by those ovarian hormones.
And the phase we're focused on now, the secretory or progestational phase, it starts about 2 or 3 days after ovulation. Perfect timing for the embryo's arrival.
Exactly. Progesterone, working together with estrogen, causes really profound changes in the endometrium. The uterine glands become highly coiled and start secreting this rich, nutrient-packed fluid. The arteries supplying the lining also become extremely coiled, like little springs.
Making the tissue.
The whole tissue becomes thick and succulent, is the term often used. It's creating the ideal receptive environment for an embryo to implant.
Wow.
At this peak secretory stage, you can actually see 3 distinct layers in the endometrium.
3 layers.
Yes. A superficial, compact layer, a thicker, intermediate, spongy layer, and then the thin, deepest basal layer.
Compact, spongy, basal.
Yep.
Okay. Now, the fate of this corpus luteum hangs in the balance. It dictates whether the cycle moves towards pregnancy or resets with menstruation. Let's take the no fertilization scenario first.
Right. If fertilization doesn't happen, the corpus luteum is essentially programmed to self-destruct. It reaches its peak development around 9 days after ovulation.
9 days.
But then it rapidly degenerates in a process called luteolysis. The lutein cells die off, the structure shrinks, and it gets replaced by fibrous scar tissue, this time called the corpus albicans, meaning white body.
Corpus albicans.
And the consequence of this degeneration is a sharp drop in both progesterone and estrogen levels. This hormonal withdrawal causes the coiled spiral arteries in the endometrium to constrict, cutting off blood supply.
Leading to
the breakdown and shedding of the top two layers, the compact and spongy layers. That shedding is menstrual bleeding.
But the basal layer, the deepest one, stays behind.
Correct. The basal layer has its own separate blood supply, the basal arteries, and it's not shed. It remains as the regenerative layer. It's from this basal layer that the endometrium will rebuild itself in the next cycle, starting the proliferative phase all over again.
Okay. Now for the scenario that changes everything. Fertilization does occur. This is where the tiny developing embryo actually takes control of the mother's endocrine system almost immediately.
It's amazing. If fertilization happens, the degeneration of the corpus luteum is actively prevented. The rescue signal is a hormone called human chorionic gonadotropin, or HCG.
HCG, that's the pregnancy test hormone?
That's the one. It's secreted by the developing embryo itself, specifically from a layer called the syncytiotrophoblast, which we'll get to.
Okay.
Under the influence of HCG, the corpus luteum doesn't just survive, it actually grows significantly. It becomes the corpus luteum of pregnancy, or corpus luteum graviditatis. It can get quite large, sometimes up to half the size of the entire ovary.
Wow. And this structure is absolutely essential for the early pregnancy.
It's non-negotiable. It sustains the pregnancy by pumping out high levels of progesterone. Progesterone maintains that secretory state of the endometrium, preventing menstruation and supporting the embryo.
How long does it keep this up?
It maintains this crucial role until about the end of the 4th month of gestation.
4 months.
After that point, the placenta, specifically its trophoblastic component, has developed enough to take over as the primary source of progesterone production. This time frame is really important clinically.
Why is that?
Because if the corpus luteum of pregnancy is removed, say during surgery before the end of the 4th month, it almost invariably leads to a spontaneous abortion, a miscarriage. The placenta simply isn't ready to provide enough hormonal support before then.
All right, the uterine environment is perfectly primed by progesterone, thanks to the corpus luteum, or the corpus luteum of pregnancy. The oocyte is waiting in the uterine tube. Fertilization itself usually happens in the ampullary region, that widest part of the tube, and typically within 24 hours of ovulation.
That's the optimal window.
Now, the journey for the sperm to get there. It's an incredible biological filter. Millions start, few arrive.
It's a massive attrition event, absolutely.
Yeah.
You might have hundreds of millions of spermatozoa deposited in the vagina, but estimates suggest only about 1% even make it into the cervix.
Only 1%.
And their movement up isn't primarily by their own swimming power, at least not initially. It's largely driven by muscular contractions of the uterus and the uterine tubes, pulling them upwards.
So they get an assisted passage.
Yes, a rapid assisted transport. This means the journey from cervix to tube can, remarkably, be completed in as little as 30 minutes, though it can also take up to 6 days for some to arrive.
30 minutes. But here's the big paradox, right? The sperm that arrive in 30 minutes are actually useless. They aren't ready to fertilize yet.
That's the critical biological delay built right into the system. Sperm absolutely must undergo 2 essential conditioning steps before they can fertilize an egg. These are capacitation and the acrosome reaction.
Capacitation and acrosome reaction. Let's start with capacitation.
Capacitation is a period of conditioning that happens within the female reproductive tract. In humans, it takes approximately 7 hours.
7 hours. Where does it happen?
Mostly occurs in the uterine tube itself, involving interactions between the sperm and the tube's epithelial lining. During this time, a kind of protective glycoprotein coat and some seminal plasma proteins are removed from the sperm's plasma membrane, specifically over the acrosomal region at the head.
So it's like stripping off armor to reveal the weapons underneath.
That's a fair analogy. And only capacitated sperm that have undergone this 7-hour process are actually capable of passing through the corona radiata cells surrounding the oocyte.
So the sperm that arrive too early just have to wait, or they fail.
Yeah.
This waiting period ensures only the fully prepped, perhaps higher quality sperm get a chance.
It seems to be a crucial selection mechanism, yes. Capacitation readies them. Then comes the second step, the acrosome reaction.
Okay, what triggers that?
This happens after a capacitated sperm successfully binds to the zona pellucida, that glycoprotein shell around the oocyte. The binding itself, particularly to a specific zona protein called ZP3, induces the acrosome reaction.
ZP3.
The reaction involves the outer membrane of the acrosome fusing with the sperm's plasma membrane. This creates openings, releasing the powerful enzymes stored inside the acrosome, things like acrosin and other trypsin-like substances.
Enzymes needed to drill through the zona.
Exactly. They need those enzymes to penetrate the zona pellucida layer.
Okay, sperm are now capacitated and have undergone the acrosome reaction. We could break down fertilization into 3 distinct phases of penetration. Phase 1, getting past the outer layer, the corona radiata.
Right. Phase 1, penetration of the corona radiata. Those 300 to 500 capacitated sperm that actually make it to the oocyte can navigate this layer relatively freely, probably helped by enzymes they release, and the general dispersal of the cumulus cells.
Only 300 to 500 make it that far, out of millions.
That's the estimate. And while only one will fertilize, the presence of the others might be important. Maybe they cumulatively help clear the path by releasing enzymes.
A group effort for the final assault. Okay, phase 2. This is the big barrier penetrating the zona pellucida. This is where the biological security system really kicks in, ensuring species specificity and, crucially, preventing polyspermy.
Absolutely critical. Polyspermy fertilization by more than one sperm is lethal in humans. The zona pellucida is this tough, species-specific glycoprotein shell. Binding to ZP3 triggers the acrosome reaction, releasing enzymes to penetrate. But the moment the head of the first successful sperm makes contact with the oocyte's own plasma membrane,
The egg reacts instantly.
The oocyte releases enzymes from specialized storage sacs called cortical granules, which lie just beneath its membrane. These enzymes flood out and cause rapid, profound changes to the zona pellucida itself. This is the zona reaction.
The zona reaction, what does it do?
It alters the structure and the chemical properties of the zona. Specifically, it inactivates those ZP3 receptor sites. So no more sperm can bind effectively, and it makes the zona itself impenetrable to any other sperm that might be trying to get through. It's the definitive, instantaneous block to polyspermy.
A biological deadbolt.
It is.
Amazing. Okay, the zona is breached by one sperm. Phase 3 is the actual physical merging the fusion of the sperm and oocyte membranes. How does that initial contact and fusion happen at the molecular level?
It involves specific molecular handshakes. There are proteins called integrins on the oocyte membrane that interact with their specific partners, ligands called disintegrins, which are found on the sperm surface.
Integrins and disintegrins.
It mediates the initial adhesion. Then, because the plasma membrane covering the very tip of the sperm head, the acrosomal cap, was actually lost during the acrosome reaction,
Right.
the actual fusion happens between the oocyte membrane and the membrane that covers the posterior region of the sperm head.
Okay, so not the very tip, but just behind it. And as the sperm enters, what parts actually go in? Just the head?
No. In humans, the pattern is that both the head, containing the nucleus, and the tail of the spermatozoon enter the oocyte cytoplasm.
The whole thing goes in.
The whole sperm cell, essentially. But its own plasma membrane gets left behind, fused with the oocyte surface.
Okay. The moment that entry happens, the egg responds immediately. You said 3 dramatic ways.
Yes. 3 simultaneous responses kick off. The first 2 are about defense and getting the egg's nucleus ready.
Defense and nuclear prep.
Response 1 is the final full activation of those cortical and zona reactions we just talked about. It makes absolutely sure the oocyte membrane is impenetrable and the zona is permanently altered. No more sperm allowed.
Got it. Defense locked down.
Response 2 is the resumption, and this time, the completion of the second meiotic division. Remember, it was arrested in metaphase.
Right, back before ovulation.
Sperm entry triggers it to finish. This produces the tiny second polar body, which gets hardly any cytoplasm, and the definitive oocyte. The oocyte's chromosomes 22 autosomes plus an X then decondense to form the female pronucleus.
The female pronucleus. Okay.
Yeah.
And the third response, you mentioned metabolic activation.
Yes, the third response is the metabolic activation of the egg. This is the true trigger, the kickstart for all the cellular and molecular events of early embryogenesis. It's the signal that essentially tells the egg, okay, let's go, develop.
And this activating factor comes from the sperm.
It's believed to be carried into the cytoplasm by the spermatozoon itself. This activation is absolutely vital because without fertilization and this activation, the oocyte would just degenerate within about 24 hours.
Okay, so now inside the fertilized egg, the zygote, we have the female pronucleus and the sperm nucleus swells up to form the male pronucleus. The tail degenerates.
Correct. You have these 2 separate haploid nuclei, the male and female pronuclei. Morphologically, they look pretty much identical at this stage.
But before they can combine their chromosomes for the first division, they have to do something crucial.
Absolutely crucial. Both the male and the female pronuclei, even though they are haploid, containing only 23 chromosomes each, must replicate their DNA.
They double their DNA content first. Why?
Think about it. If they didn't replicate their DNA before the first division, when the zygote divides into two cells, each daughter cell would only get half the normal amount of DNA needed for a diploid cell. That would be non-viable. So they each synthesize DNA, going from 23 single chromosomes, 1N, 1C, to 23 double chromosomes, 1 and 2C.
Ah, I see. So each pronucleus now has the right amount of DNA for a future diploid cell, just packaged separately for a moment.
Exactly. Once DNA replication is complete in both pronuclei, their nuclear envelopes break down. The chromosomes condense, and now you have the 23 double chromosomes from the father and the 23 double chromosomes from the mother arranging themselves together on the first mitotic spindle.
Ready for the first division.
Right. The chromosomes split longitudinally at their centromeres, and the sister chromatids move to opposite poles. This ensures that each of the two resulting daughter cells receives the normal diploid number of chromosomes, 46 single chromosomes. Then a furrow appears, dividing the cytoplasm.
And we have the two-cell stage, the zygote.
The two-cell stage zygote. And that single event of fertilization has achieved three absolutely fundamental results that define this new individual.
Okay, what are the three main results?
First, obviously, the restoration of the diploid number of chromosomes, 46 total. This unique combination of maternal and paternal genes sets the genetic blueprint.
Right, diploid number restored.
Second, the chromosomal sex of the individual is determined right at this moment. It depends entirely on the sperm. If the sperm carried an X chromosome, the zygote is XX, female. If it carried a Y chromosome, the zygote is XY, male.
Sex determined. And third,
and vital for everything that follows, fertilization initiates cleavage, that rapid sequence of cell divisions that starts immediately. Without fertilization, as we said, the oocyte just degenerates. Fertilization kickstarts the developmental program, #CPH2SEC, cleavage, compaction, and blastocyst formation.
Okay, the first week now really shifts into high gear with cleavage, this series of rapid mitotic divisions. But because the embryo is still trapped inside that original shell, the zona pellucida.
Right, the zone is still there.
The resulting cells, the blastomeres, they actually have to get smaller with each division, right? The overall size doesn't increase yet.
Exactly. The total volume stays about the same, but the number of cells increases. So each individual blastomere becomes progressively smaller. And there's a pretty precise, rapid timeline for this.
What's the timing like?
The two-cell stage is typically reached about 30 hours after fertilization, then the four-cell stage around 40 hours post-fertilization.
Okay, 30 hours, 40 hours.
But up until about the 8-cell stage, the blastomeres form a sort of loose, relatively unstructured clump inside the zona. And this brings us to a really essential process called compaction.
Compaction. This sounds important. It's the first real sign of organization, of cells starting to know their fate. It turns that loose ball into something more tightly knit.
That's exactly what it is. It's the first major morphological sign of differentiation. After the third cleavage, around the 8-cell stage, the blastomeres suddenly change their behavior. They maximize their contact with each other, flattening down.
How do they stick together so tightly?
They start forming specialized cell-to-cell junctions, particularly tight junctions, around their outer surfaces. These junctions seal the spaces between the outer cells and hold them together so effectively that, from the outside, the individual cell outlines become quite indistinct, hence the term compaction.
And this physical process achieves the first crucial segregation of cells.
Yes. Compaction physically separates the cells that are now truly on the inside of the clump from those on the outside. The inner cells maintain extensive communication with each other through gap junctions.
Okay, so T-junctions seal the outside, gap junctions connect the inside.
Right. And the segregation is fundamental for determining cell fate.
So in just about 3 days, the embryo has basically started deciding. U cells on the outside will become the placenta, and U cells on the inside will become the actual baby. By the time it reaches 16 cells, we give it a specific name.
Yes, around 3 days after fertilization, when the compacted embryo consists of about 16 cells, it's called the morula. Comes from the Latin for mulberry because it kind of resembles one.
Morula. 16 cells, day 3.
And at this morula stage, those two cell populations are clearly established. The cluster of internal cells is now officially called the inner cell mass. This is destined to give rise to all the tissues of the embryo proper, the fetus itself.
Inner cell mass becomes the embryo. And the outside.
The surrounding layer of external cells is now called the outer cell mass. This will form the trophoblast, which is the primary layer that will later contribute extensively to the formation of the placenta.
Inner cell mass embryo.
Yep.
Outer cell mass trophoblast placenta.
Got it.
Now, the morula doesn't stay in the tube, does it? It moves into the uterus.
Right. Around day 4 after fertilization, the morula completes its journey down the uterine tube and enters the main uterine cavity. And this is where the next critical step begins. Blastocyst formation. It's driven by fluid.
Fluid from the uterus.
Exactly. Fluid from the uterine cavity starts to seep through the zona pellucida. It's still present but permeable, and begins to accumulate in the spaces between the cells of the inner cell mass. These small fluid-filled pockets rapidly merge or coalesce to form one single large fluid-filled cavity. This cavity is called the blastocele.
Blastocele.
And once this cavity forms, the entire structure is now defined as a blastocyst.
So morula becomes blastocyst when the fluid cavity forms.
Correct. And the structure is now clearly organized. The inner cell mass, now called the embryoblast, is pushed off to one side, clustered at one pole of the sphere. And the outer cell mass, the trophoblast, has flattened out to form the thin epithelial wall of the blastocyst surrounding the blastocele.
Embryoblast at one pole, trophoblast forming the wall. Okay, the blastocyst is ready for implantation, but there's one last barrier that Charlotte's been inside the whole time, the zona pellucida.
Right, the zona pellucida has served its purpose. It prevented premature implantation in the tube and kept the blastomeres together. But now, for implantation to occur, it has to go.
Why does it disappear?
It degenerates and disappears, often referred to as hatching, usually around the end of the 4th day or early on the 5th day.
Hatching, like a chick from an egg.
Similar idea. Its removal allows the blastocyst to finally expand and, more importantly, to make direct contact with the uterine wall lining, the endometrium.
And implantation begins when?
The source material pinpoints the initiation of implantation to about the 6th day after fertilization. It starts when the trophoblast cells, situated directly over the embryoblast pole, begin to actively penetrate, squeezing between the epithelial cells of the uterine mucosa.
So it's the pole with the inner cell mass that leads the invasion.
Yes, that pole attaches first. And that initial attachment isn't just passive sticking. It's a very specific molecular process, a handshake, as you called it earlier.
What molecules are involved in this first grab?
The very initial, somewhat loose attachment, think of it like Velcro catching, is mediated by molecules called L-selectins. These are found on the surface of the trophoblast cells.
L-selectins on the trophoblast.
And they bind specifically to carbohydrate receptors present on the surface of the uterine epithelial cells. It's actually quite analogous to how white blood cells, leukocytes, are captured from the bloodstream by the lining of blood vessels during inflammation.
A selectin-mediated capture. Okay, so it's loosely attached. How does it transition from just sticking to actually invading? What drives the deeper connection and migration?
That requires a different set of molecular players. Integrins.
Integrins again.
Yes. The trophoblast cells express various types of integrin receptors on their surface. These integrins then bind to molecules within the uterine endometrium's extracellular matrix, the stuff between the cells.
Like what matrix molecules?
Specifically, binding to laminin seems to promote strong, stable attachment. Binding to another molecule, fibronectin, appears to actively stimulate the trophoblast cells to migrate and invade deeper into the uterine tissue.
Laminin for sticking, fibronectin for moving.
That seems to be the idea. So implantation isn't just the embryo pushing its way in. It's a dynamic, coordinated process involving mutual signaling and action between the trophoblast and the receptive endometrial lining.
It's amazing. Even as the blastocyst is just starting to anchor itself around day 6, inside that little ball, the inner cell mass, the future embryo, is already busy organizing itself into layers and setting up its fundamental body plan, its axis. This internal differentiation gets a push from growth factors.
Absolutely. Even at the early blastocyst stage, perhaps just before implantation starts, the cells within the embryoblast differentiate into 2 distinct layers. These are the epiblast and the hypoblast.
Epiblast and hypoblast.
And this differentiation process is known to be influenced by signaling molecules from the fibroblast growth factor, or FGF, family.
Okay, FGF helps make epi and hypo. And how are they arranged? Does their position mean something?
It absolutely does. Their physical positioning establishes the very first body axis, the dorsal ventral axis, or back to belly. Right around the time of implantation, the epiblast cells arrange themselves to form the upper or dorsal layer. This layer will eventually give rise to the backside of the embryo. The hypoblast cells form the lower or ventral layer, which is right next to the blastocell cavity. This establishes the future belly side.
Epi on top, dorsal back, hypo below, ventral belly. Simple enough, but setting up the head-to-tail axis, the cranial-caudal axis, that sounds more complex. Our sources say it involves active signaling and even inhibition to make sure the head forms in the right place. It involves a special group of hypoblast cells.
Yes, this is a fascinating and critical process for ensuring the body plan develops correctly. A specific small population of cells within the hypoblast layer gets designated to become the anterior visceral endoderm, or AVE.
Anterior visceral endoderm, AVE.
These AVE cells then undertake a directed migration. They move specifically to the location within the embryoblast that is destined to become the cranial and the future head end of the embryo.
Okay, the AVE cells mark the spot for the head. But what's their actual job there? Are they sending out build the head here signals, or is it more like stopping something else from happening there?
It's fundamentally an inhibition mechanism. The AVE cells are crucial because they secrete specific signaling molecules that block the action of another powerful signaling molecule called nodal. These blocking molecules are nodal antagonists.
Nodal antagonists, like specific chemicals.
Yeah. Yes, the two main ones mentioned in our sources are named Cerberus and Lefty-1.
Cerberus and Lefty-1. Great names.
They are. So think of Cerberus and Lefty-1 secreted by the AVE at the future head end as biological signs saying, nodal is blocked here, this area is designated cranial.
Okay, so they inhibit nodal activity at the head end. What does nodal do where it's not inhibited?
Exactly. In the region away from the AVE, where these inhibitors aren't present, the nodal signal is free to act. And nodal's job, where it's active, is to establish the primitive streak.
The primitive streak, that's the structure that initiates gastrulation, right? Forming the germ layers.
Precisely. The primitive streak forms at the opposite end from the AVE, the caudal or tail end. Nodal signaling there initiates gastrulation and the formation of the mesoderm and endoderm layers. So this elegant interplay between the AVE establishing an inhibitory zone at one end and nodal acting freely at the other sets up the fundamental cranial-caudal axis.
And this all happens incredibly fast, right around day 5.5 to 6, establishing the basic body blueprint just as implantation gets underway.
It's a remarkable feat of timing and molecular coordination. #tag8, clinical applications and challenges.
Okay, we've meticulously followed the perfect sequence of events for the first week, but now we need to shift focus to the clinical reality when things don't go according to plan. Let's start with infertility, which the sources state affects a significant number of couples, maybe 14 to 15%. We need to look at both male and female factors.
Right. Starting with male factors, the most common issues relate to sperm count and sperm motility.
Quantity and quality of movement.
Exactly. A typical ejaculate might be 2 to 6 milliliters and could contain, up to 100 million sperm per milliliter. But the functional thresholds for fertility are lower, though still critical.
What are those thresholds?
Generally, fertility often requires a concentration of at least 20 million sperm per milliliter or a total count of at least 50 million sperm in the entire ejaculate. Falling below those numbers, a condition called oligozoospermia makes natural fertilization much harder.
Oligozoospermia, low sperm count.
And even if the count is okay, poor motility sperm that don't swim well or straight is also a major barrier. They just can't make that long journey effectively.
Okay, those are key male factors. What about female infertility? The potential causes seem more diverse, impacting different stages.
That's true. Female factors can arise at the level of the ovary, the cervix, or the uterine tubes. One major mechanical issue is blocked tubes, occluded uterine tubes.
Blocked fallopian tubes. What causes that?
Often, it's a consequence of pelvic inflammatory disease, PID, which is usually caused by bacterial infections, sometimes sexually transmitted ones. The inflammation can scar and block the tubes.
And if the tubes are blocked,
then sperm and oocyte simply can't meet in the ampulla where fertilization normally occurs. It's a physical impossibility.
Makes sense. What else on the female side?
Other issues can include hostile cervical mucus. Remember how estrogen needs to thin it? If that doesn't happen properly, the mucus remains thick and impenetrable to sperm.
Right.
There can also be immunological problems where a woman's immune system actually develops antibodies against sperm, treating them as foreign invaders. And of course, a fundamental issue is the complete absence of ovulation, or anovulation.
Anovulation. So for women who aren't ovulating regularly, or perhaps for IVF preparation, drugs like clomiphene citrate are often used. We mentioned it earlier. How does Clomid actually work?
Clomiphene citrate, or Clomid, is typically taken early in the menstrual cycle. Its main action is to effectively increase the concentration and the impact of follicle-stimulating hormone, FSH.
Boosting FSH.
Right. By enhancing the FSH signal, it encourages better follicle development and increases the likelihood of triggering ovulation. So it's used both to induce ovulation in women with irregular cycles and, as you said, it's a standard part of preparing women for egg retrieval during in vitro fertilization, IVF, to maximize the number of mature oocytes collected.
Okay, which leads us directly into assisted reproductive technology, or RT. The sources note that RT accounts for maybe 1 to 2% of all births now in the U.S. That's significant. But we also need to address the clinical consensus about the associated risks compared to natural conception.
Yes, RTs involves manipulating sperm, eggs, and early embryos outside the body, and the data consistently shows higher rates of certain adverse outcomes.
Notice.
These include increased rates of prematurity babies born before 37 weeks gestation. Also, low birth weight, which is defined as less than 2,500 grams or about 5.5 pounds, and even very low birth weight, under 1,500 grams.
Preterm birth, low birth weight, anything else?
There's also a documented increase in the incidence of certain types of birth defects, even when the procedures seem routine.
Now, traditionally, a lot of this increased risk was thought to be due to the common practice in RT of transferring multiple embryos at once, which obviously leads to more twins, triplets, etc., and multiple births inherently carry higher risks. Does the source material clarify if the risk is just from multiples, or is there something about the RT process itself?
That's a really crucial distinction, and the sources do address it. While it's undeniable that a large proportion of the adverse outcomes are linked to the high rate of multiple births, which often happens because clinics transfer, say, 4 or 5 embryos to try and boost the chances of at least one implanting.
Right.
More recent, carefully controlled studies indicate that even singleton births, just one baby resulting from ARC procedures, still show a measurable increase in the risk of preterm birth and certain malformations compared to naturally conceived singletons.
So even with just one baby, there's still some increased risk.
It suggests that either the process of in vitro culture and handling itself, or perhaps the underlying infertility factors and the parents that necessitated RT in the first place, might carry some inherent non-negligible risk beyond just the issue of multiple gestation.
That's important to understand. Okay, let's quickly detail the 2 main RT methods mentioned. First, standard IVF and embryo transfer.
Right. In standard IVF, as we discussed, follicle growth is stimulated using gonadotropins. Oocytes are retrieved surgically just before they would naturally ovulate, usually when they're in late meiosis the first.
Okay.
They're then placed in a special culture medium in the lab, and prepared capacitated sperm are added to achieve fertilization in the glass. That's what in vitro means.
Fertilization in a dish.
Exactly. The resulting fertilized eggs, the embryos, are then carefully monitored as they divide. Typically, they're cultured until they reach the 8-cell stage, which takes about 3 days. Then, 1 or more of these embryos are transferred via a catheter into the woman's uterus.
And success rates vary a lot.
Highly dependent on factors like maternal age dropping off quite significantly after age 40.
Okay, that's standard IVF. Now contrast that with intracytoplasmic sperm injection, or ICSI.
Right.
This is used more for severe male infertility factors, right? Like very low sperm count, oligospermia, or even no live sperm in the ejaculate, azoospermia. ICSI seems to bypass many of the natural steps sperm normally take.
It's a much more direct intervention, yes. In ICSI, a single individual sperm, which can sometimes be harvested directly from the man's epididymis or testes if none are in the ejaculate, is selected.
Just one sperm.
One sperm is picked up with a very fine needle and is manually injected directly into the cytoplasm of the retrieved egg.
Wow, straight into the egg.
It completely bypasses all the natural barriers and selection processes. The need for capacitation over 7 hours, the acrosome reaction to penetrate the zona, the whole journey through the female tract, even binding to the zona, it circumvents all of it.
While that offers a solution for severe male infertility, does bypassing all that natural selection potentially contribute to the risks we were just talking about? Those natural steps seem like important quality control checks.
That's precisely the concern many experts raise. By mechanically circumventing these evolved natural selective pressures steps that presumably weed out weaker or abnormal sperm, are we potentially allowing sperm with subtle defects, maybe genetic ones, to fertilize the egg when they wouldn't have succeeded naturally?
Does the source material link ICSI to specific risks?
Yes, it points to a specific increased risk associated with ICSI for male fetuses inheriting certain Y chromosome deletions from their fathers, which might have been the cause of the infertility in the first place. Also, some studies suggest ICSI might be associated with a slightly higher incidence of certain birth defects compared even to standard IVF, though the data is complex. It certainly raises questions about the long-term implications of bypassing nature's screening.
It really highlights the importance of those natural processes. Which brings us back to something the sources emphasize, the incredibly high rate of natural pregnancy loss, or spontaneous abortion. Up to 50% of all pregnancies may end naturally, often before a woman even knows she's pregnant. This seems like evolution's own very stringent quality control.
It is a sobering statistic, but it reflects a crucial biological function. About half of those very early, often unrecognized spontaneous losses are directly caused by major chromosomal abnormalities in the embryo, things like having the wrong number of chromosomes, aneuploidy, or large structural rearrangements.
So nature has this built-in screening mechanism.
Exactly. This high rate of natural loss acts as a powerful screening mechanism, effectively purging the reproductive system of a large proportion of embryos that are genetically non-viable or severely defective. The sources estimate that without this rapid early screening, the incidence of congenital malformations seen in babies born alive would jump dramatically from the current background rate of about 2 to 3% up to maybe 12%.
Wow, that's a huge difference. So, given this natural screening, what's the role of modern genetic screening techniques used with IVF, like pre-implantation genetic diagnosis, PGD, where a single cell, a blastomere, is removed from an early embryo and its DNA is tested? Is it just mimicking nature or doing something different?
It's essentially trying to front-load the screening process and make it more specific. PGD combines IVF with molecular tools like the polymerase chain reaction, PCR, to amplify the tiny amount of DNA from that single blastomere.
Okay.
This allows screening for specific known genetic defects like the mutations causing cystic fibrosis or Huntington's disease, or for chromosomal abnormalities before the embryo is even transferred to the uterus.
So it allows selection based on known risks.
Right. It lets couples at high risk for certain inherited disorders avoid transferring an affected embryo. In that sense, it's different from the natural system, which seems to screen more broadly for overall viability, leading to that high general loss rate. PGD targets known issues identified through advances like the Human Genome Project. It's becoming increasingly common, especially for older couples or those with known genetic risks.
Okay, let's shift gears slightly now to stem cell technology. This obviously leverages the very earliest stages of development we've been discussing. We need to be really clear on the scientific and also the ethical distinctions between embryonic stem cells, ES cells, and adult stem cells and how they're obtained.
Absolutely. Embryonic stem cells, ES cells, are derived exclusively from the inner cell mass of the blastocyst. Remember, that's the part destined to become the embryo proper.
Right, the embryoblast.
ES cells have this remarkable property called pluripotency.
Pluripotent? Meaning
They have the potential to differentiate into virtually any type of cell or tissue in the entire body: muscle, nerve, liver, pancreas, anything. This incredible flexibility is why they hold such immense therapeutic promise for treating degenerative diseases like diabetes, Alzheimer's, Parkinson's, or repairing damage from things like heart attacks or spinal cord injuries.
huge potential. But the methods for getting these pluripotent cells are where the ethical and practical issues arise. Let's distinguish the two main approaches mentioned in the sources.
Okay, the first way ES cells have been obtained is sometimes loosely termed reproductive cloning in the context of sourcing cells, though that term is often misused. It involves using surplus embryos that were created during standard IVF procedures for infertility treatment, but are no longer needed for that purpose.
Using leftover IVF embryos.
Right. The inner cell mass is isolated from these blastocysts, and ES cell lines are established. The primary ethical concern here, for many people, revolves around the use and destruction of a potential human life, an embryo. From a practical medical standpoint, there's also the significant risk of immune rejection if these cells are transplanted into a patient, because they wouldn't be genetically identical to the recipient.
Okay, ethical debate and immune rejection risk with IVF-derived ES cells. What's the second approach? It's often called therapeutic cloning, or more technically, somatic nuclear transfer, SCNT. This tries to address the immune rejection problem.
Exactly. Therapeutic cloning, or SCNT, aims to create patient-specific ES cells. Here's how it works. You take the nucleus out of an adult body cell, say a skin cell, from the patient who needs the treatment.
Okay, get the nucleus from the patient's cell.
Then you take an unfertilized human egg cell, an oocyte, and remove its own nucleus. This is called an enucleated oocyte.
An empty egg cell, basically.
You then introduce the nucleus from the patient's adult cell into this enucleated oocyte. This reconstructed cell is then stimulated chemically or electrically to start dividing as if it had been fertilized. It develops in vitro into a blastocyst.
A blastocyst genetically identical to the patient.
Precisely. The inner cell mass is then harvested from this blastocyst to create ES cell lines. Because these ES cells contain the patient's own DNA, they are genetically compatible, theoretically eliminating the problem of immune rejection upon transplantation.
And ethically.
Ethically, SCNT is often considered less controversial by some because it does not involve the fertilization of an egg by a sperm, nor does it use embryos originally created for reproductive purposes. However, it still involves creating and destroying a blastocyst, which remains an ethical concern for others.
Okay, pluripotent ES cells with huge potential but sourcing challenges. Now contrast those with adult stem cells. They exist in our bodies already, but they're different.
Yes, adult stem cells are found in various tissues in the body even after development is complete. Think bone marrow, fat tissue, even the brain. However, they are classified as multipotent, not pluripotent.
Multipotent, meaning less flexible.
Much less flexible. They generally have a restricted differentiation ability. They can typically only form a limited range of cell types related to the tissue they came from. For example, bone marrow stem cells primarily make blood cells.
So, less potential than ES cells.
Generally, yes, although research is constantly pushing those boundaries. For instance, the source notes experiments where adult stem cells from rat brains were successfully used to cure Parkinson's-like symptoms in other rats, which is very promising.
But there are drawbacks to using adult stem cells.
Two main practical disadvantages currently. First, they're often very scarce within the tissue, making them difficult to find and isolate. Second, they tend to divide much more slowly in culture compared to ES cells, which makes it challenging to grow enough of them for large-scale experiments or clinical treatments. Research is ongoing to overcome these limitations.
Okay, finally, for this clinical section, let's quickly cover the mechanisms of action for the main contraceptive methods outlined in our sources.
Yeah.
How do they work to prevent pregnancy?
Right. Hormonal contraceptives, things like birth control pills, patches, rings, injections, usually contain either a combination of synthetic estrogen and progestin or just progestin alone. They work primarily through three interconnected mechanisms.
3 ways.
First, and most importantly, they inhibit ovulation. The hormones provide negative feedback to the hypothalamus and pituitary, suppressing the release of GnRH, FSH, and particularly that critical LH surge. No LH surge, no ovulation.
Okay, start ovulation. What else?
Second, they cause changes in the uterine lining, the endometrium, making it thinner and less receptive to implantation, just in case ovulation somehow still occurred and fertilization happened.
Less welcoming lining. And third,
particularly the progestin component, thickens the cervical mucus, making it viscous and impenetrable. This forms a physical barrier that blocks sperm from entering the uterus in the first place.
okay, hormonal methods hit ovulation, lining, and mucus. What about intrauterine devices, IUDs?
IUDs are small, usually T-shaped devices placed inside the uterus. There are two main types. Hormonal IUDs release a small amount of progestin locally within the uterus. This primarily works by thickening the cervical mucus like the pills and may also affect sperm viability and the uterine lining.
Okay, hormonal IUD is like local progestin. What about the copper IUD? No hormones there.
Correct. The copper IUD releases copper ions into the uterine cavity. Copper is toxic to sperm. It acts as a spermicide, preventing fertilization from happening. It also seems to create a sterile, inflammatory response in the endometrium that makes implantation very difficult, if not impossible, should fertilization somehow occur. So it works primarily by preventing fertilization and secondarily by preventing implantation.
Copper is spermicidal and anti-implantation. Got it. Lastly, emergency contraceptive pills, ECPs, sometimes called the morning after pill. How do they work?
ECPs are designed for use after unprotected intercourse, but before pregnancy is established. The most common type, like Plan B, contains a high dose of progestin, levonorgestrel. Taken within, say, 72 to 120 hours, its main effect is believed to be delaying or inhibiting ovulation if it hasn't already occurred.
So it primarily stops the egg from being released.
That's thought to be the primary mechanism, yes. Okay. There's less evidence it significantly prevents implantation if fertilization has already happened. Then you have other types of ECPs that use different agents, like ulipristal acetate, brand name Ella, which is a selective progesterone receptor modulator, also primarily working by delaying ovulation, perhaps more effectively closer to the LH surge. And then there's mifepristone, RU486, which is a potent anti-progesterone drug.
Mifepristone. That's also used for medical abortions, right?
Exactly. It's important to note the distinction. Mifepristone can function as an emergency contraceptive if taken before implantation by disrupting the hormonal environment needed for potential implantation. However, it is also highly effective as an abortifacient, causing the termination of an established pregnancy if taken after implantation has already occurred, because it blocks the progesterone needed to maintain the pregnancy. So its action depends heavily on the timing relative to fertilization and implantation. #IX. Outro and final thought.
Wow. Okay, this has been an incredibly dense, but I think really clarifying journey through that first week. I feel like we really have approached mastery of this critical period. We've traced it all the way from that initial hypothalamic signal, the GnRH,
Right, the starting gun.
all the way through the hormonal cascade, the follicle selection by FSH, the power of the LH surge, the intricate steps of fertilization, including that 7-hour capacitation wait, and the crucial zonal reaction.
Can't forget the zonal reaction.
Absolutely. And then on to cleavage. The vital step of compaction, creating the inner and outer cell masses, blastocyst formation, hatching, and finally that molecular anchoring using L-selectins and integrins around day 6, and even establishing the body axis with the AVE and nodal signaling.
We really did cover the essential transitions, understanding that selection pressure from FSH, the controlled explosion of ovulation via LH, that mandatory delay for capacitation ensuring sperm readiness, and the absolute necessity of the zonal reaction blocking polyspermy. Those are fundamental. And then we saw how disruptions in these incredibly precise micro-events lead directly to major clinical challenges like infertility, and how our attempts to intervene with art, while often successful, carry their own complexities and risks. Plus, the huge potential and ethical considerations surrounding stem cell technology, which arises directly from these very early stages.
What really stands out to me looking back is the almost ruthless optimization of the whole system. But it seems optimized for quality, often at the expense of sheer numbers or early survival. Particularly evident in that astonishingly high natural loss rate, maybe 50%.
It truly is the ultimate biological quality control system, operating right from the start. And that point you raised earlier about the timing conflict is fascinating.
The conflict between the potential for rapid 30-minute sperm transport versus the mandatory 7-hour wait for capacitation.
Exactly. Evolution seems to have deliberately built in this critical, unavoidable delay. It forces the sperm that arrive quickly to wait, ensuring they undergo the necessary molecular changes, the conditioning, before they're even allowed to attempt fertilization. Speed alone isn't enough. Preparedness is key.
So perhaps here's our final provocative thought for you, our listeners, to mull over. Why this specific design? Why did evolution favor a system where the mechanisms for rapid transport seem almost pitted against the mandatory lengthy preparation time needed for success?
What does this inherent tension, this high-stakes race, where conditioned quality ultimately trumps raw speed, tell us about the absolute minimum biological criteria needed to generate a viable human zygote?
And how might that very conflict, that requirement for perfect timing between arrival and readiness, contribute to the inherent vulnerability and the high natural loss rate observed in this incredibly critical first week of life?
That tension, that balance between getting there and being ready, you could argue it's the single most critical filter in the entire process of human conception.
An intricate, high-stakes biological dance indeed. Thank you so much for joining us in this very deep dive.
A pleasure.
We hope you found it illuminating, and we encourage you to continue exploring the incredible complexities of early biology. Until next time.